Hello and welcome to your video lecture in section 17.2, where we are going to talk about hormones. So our hormones serve as chemical messengers, and they are released by endocrine cells and glands into our bloodstream, and they are transported throughout the body and will eventually reach a target cell. They are going to regulate the metabolic functions and activities of other cells in our body. And we have three major classifications for our hormones. We will have our amino acid derivatives, our peptide hormones, and lipid derivatives. So let's talk about each of these classifications first, and then we will revisit what is going on in this figure here. So first up is our amino acid derivatives. This means the hormone comes from a single amino acid. So examples would include our thyroid hormones such as thyroxine and triiodothyronine. You may also know this as T4 and T3. And our catecholamines that we see pictured over here, which include epinephrine and norepinephrine, as well as dopamine. And the last one we're going to learn about is melatonin. Remember, this is the hormone that helps us set our circadian rhythms and is released by our pineal gland. Next up is our peptide hormones. These are chains of amino acids, which you could see over here. So each of these circles are representing a different amino acid. And examples would include polypeptides, such as our antidiuretic hormone, which we see over here. It's made up of 9 amino acids. And oxytocin, which is also made up of 9 amino acids. But of course, we do see some differences in here as far as the specific type of amino acid that is in this chain. Other types would be our small proteins, which is insulin, made up of 51 amino acids, growth hormone, made up of 191 amino acids, and prolactin, which has 198 amino acids. Now, I'm not expecting you to memorize these numbers, but do know that these hormones are small proteins. And lastly, we have our glycoproteins, which would include our thyroid stimulating hormone, luteinizing hormone, and follicle stimulating hormone. And here you could see that the protein is folded. These are called alpha helices. So it's a much larger type of protein in comparison to our small protein hormones and polypeptides. And our last classification are our lipid derivatives. These consist of these carbon rings and side chains that are built either fully from fatty acid chains or cholesterol. And so this you could see is our chemical structure of cholesterol, whereas we see a similar look over here, but of course we've got different side chains going on and bonds. So first we have our eicosanoids, and these are built from our fatty acid chains and include leukotrienes and prostaglandins. We'll talk about this a bit more within our immune and lymphatic systems. And we have our steroid hormones, which are built from cholesterol. And these will include testosterone, estrogen, progesterone, our corticosteroids, and calcitrol. Now, how do these hormones go about entering the cell and triggering their hormone response, or the mechanism that is going to help them cause the effect that they want? First, we need to look at how our lipid-soluble hormones do this. So a steroid hormone, which we could see pictured over here, is going to directly initiate the production of proteins within the target cell. It does this by easily diffusing through the cell membrane. You could see this is happening rather quickly in this photo. And then it's going to bind to what we call a receptor hormone complex. So it could do this in the cytoplasm out here, and then enter our nucleus and bind to the target gene on the DNA. And then through the process of transcription, we are going to create a messenger RNA strand. So if you remember what transcription is, we essentially unwind DNA so we can read the nucleotides and create a messenger RNA. This serves as kind of a temporary strand in which we are going to read to create a protein. And that process where we read the mRNA strand to create a protein is called translation. And that will take place back out in the cytoplasm. So you might be wondering, well, why don't we just have translation read directly from DNA? Well, if something goes wrong, we don't want to damage our DNA, and that's why we have this temporary mRNA strand that gets read in order to create the protein. So this again is the mechanism for lipid-soluble mechanism of action. There is a video that you can click on and watch. I won't play it here just for the sake of time, but it is worth to see an animation as to how this takes place. The other mechanism is for our water-soluble hormones. Unfortunately, this cannot diffuse through the cell membrane like we saw with the lipid-soluble. So this would be maybe something to really remember for a test question, hint, hint. So our water-soluble hormone leaves the bloodstream and has to attach to a cell membrane receptor first. Let's add that in, a surface cell membrane receptor here. This receptor is then going to initiate cell signaling pathways that involve our G proteins. And you see this really well again in this animated video where some of the subunits are going to go across and our adenylyl cyclase is going to then become catalyzed and converts ATP into our cyclic AMP, which serves as our second messenger in this pathway. That is important to remember. And then the cyclic AMP activates our protein kinase, which then goes through and has a cascade effect. It's going to phosphorylate our proteins within the cytoplasm and activate proteins in the cell that can carry out the specific changes that we have for whatever hormone that it is. So just really knowing that we have a cascade effect that activates proteins is enough. Now, what are the factors affecting target cell response? We know by now that our hormones have to have receptors on their target tissues in order to work properly. So sometimes in upregulation, we can have a stimulus that can cause an increase in the synthesis of receptors for a hormone. And this would then increase our sensitivity to that hormone. So here's our hormone that makes it into the cell, and then we create that messenger RNA that is going to create more receptors that we can place on the cell membrane. For example, FSH, the follicle stimulating hormone, when that's stimulated, it is going to cause an increase in LH receptors, the luteinizing hormone. And our ovarian cells then can be more sensitive to LH, even if the concentration of LH hasn't changed. So this can help cause ovulation. That'll make a little bit more sense once we go through what FSH and LH are, but if you're already familiar with that from your prior transfer AMP courses, hopefully that resonated. With our downregulation, we can see a decrease in receptor number. So we are going to have a cell that are exposed to a hormone repeatedly, and a combination of hormones and receptors can increase the rate at which the receptor molecules become degraded. This combined form is taken into the cell through phagocytosis and broken down. So again, hormone is going to move in. We start to see that we degrade those receptors again through the process of phagocytosis. So now we are less reactive to the excess hormones because they need a receptor to bind to, and we don't have a lot. All right, what about antagonistic effects? One hormone can help inhibit the response of another, and therefore they're generating the opposite responses. So a good example of this is insulin and glucagon. We know that insulin is going to lower blood sugar. It takes the glucose from the blood and puts it into cells, while glucagon is going to help raise blood sugar. What about synergistic effects? This is when we have two hormones that have a similar effect but can help amplify a response. So in some cases, two hormones are going to be required for an adequate response. But here we see an example of estrogen and LH working together in order to get the synergistic effect that they want. We also see a similar effect when it comes to FSH and estrogen from our ovaries, and that is needed to get our maturation of the female ova or the egg. And of course, we have permissive effects when we have one hormone that is needed to activate another. So a good example of this would be renin, which renin is released from our kidney and is needed in order to convert our angiotensinogen into angiotensin 1. And then we have another enzyme that comes in called angiotensin converting enzyme, also known as ACE, ACE, that converts angiotensin 1 to angiotensin 2. And angiotensin 2 assists with one, activating secretion of another hormone known as aldosterone, as well as having certain effects such as vasoconstriction to raise blood pressure. Now let's move into feedback mechanisms. You may have learned this before. We have a negative feedback system and a positive feedback system. So some of the main differences I want you to keep in mind if you've never really kind of understood these mechanisms is that the negative feedback system is almost always, let me say, the goal of a negative feedback system is to always achieve homeostasis, okay? Whereas our positive feedback system, it is always there to amplify. And we don't have many systems in our body that normally will use positive feedback mechanism in a helpful way. Most of the time when that's going on, it's pathological. But we'll talk about some natural positive feedback loops that are okay or healthy for our body to go through. So anyway, let's focus on negative feedback system first. That is going to be a physiological response that causes a decrease in a release of a hormone. That's the most commonly used type in our body, is this negative feedback system, especially when it comes to the endocrine system. So we start to get an imbalance of something. And in this example, our hypothalamus is perceiving low concentration of glucocorticoids because we've got some sensors in our blood vessels for those glucocorticoids. So we've got a low amount of that. So in order to, our goal, again, is to reach homeostasis. So in order to increase our glucocorticoids, we want hormone release. So our hypothalamus, that we see that triangular shape over here, is going to release something known as our corticotropin releasing hormone. Now we haven't talked about the hypothalamus specifically, but it is in charge of a lot of releasing hormones and inhibiting hormones. It basically tells the pituitary gland, for the most part, what to do. So it tells the pituitary gland now, hey, I've got this releasing hormone, and so from the pituitary gland, we are going to release another hormone, which I'm surprised I don't have listed here. But that other hormone is going to be our adrenocorticotropic hormone. I know that's a really long name. We'll talk about it later on, but you can know it now as ACTH. So ACTH gets released from the anterior pituitary gland. It travels to the cortex of the medulla that sits on top of the kidney and will tell it to release our glucocorticoid hormone. That then will increase those levels in the blood. And so now that we have our homeostasis level, the negative feedback part of that is to stop or inhibit the releasing hormone from being released from the hypothalamus. We can also signal to the anterior pituitary gland to stop releasing the ACTH to go to the cortex and release glucocorticoids because we have enough in our blood. Now for our positive feedback systems. Again, this is a physiological response that causes an increase in a release of a hormone. Again, naturally it is rarely used, but we do see it in certain mechanisms. One of them is childbirth. Another one is for our blood to clot. So our baby in the womb is going to have its head push against the cervix. Here we have sensory nerves that are going to send impulses from the uterus here to the brain. Our brain then recognizes that we need the pituitary gland to secrete a hormone known as oxytocin. Oxytocin gets released through the blood. It travels to the uterus, and one of the main actions the oxytocin hormone causes is uterine contraction. So we are going to have the uterus contract. When it contracts, it pushes the baby's head against the cervix more, sending signals to the brain, causing more secretion of oxytocin. And of course, we know the contractions are getting stronger and closer together. Again, the big word here with our positive feedback loop is amplification. We are increasing and amplifying the release of that hormone until we get the desired effect. In this case, that baby's head is going to push against the cervix, sending those nerve signals to the brain, telling pituitary gland to release oxytocin until this baby comes out through the birth canal.